Hello, welcome everyone. Good evening. It's so nice to see so many of you joining us here on, I know it's a lovely sunny evening outside, so thank you for choosing to spend it indoors with us. Welcome to the second of our COVID Connections discussion events that are staged in partnership between the Wellcome Genome Campus and the COVID-19 Genomics Consortium in the UK, COG UK. We set up this programme of events to explore how genomics is helping combat the coronavirus, which has obviously impacted our lives here in the UK and around the world over the last nearly 18 months. And genomics is leading the way in how we tackle that through multiple different avenues. Tonight we'll be exploring how variations occur, what variants mean, and when they really become a cause for concern. All of our speakers have a connection to the COG UK consortium, contributing really vital work through whether that's in universities, research institutes, in clinical settings. They are working tirelessly to help us in this effort, and we'll be hearing I hope will be a fantastic kind of conversation between them, but most importantly for us, between you here joining us this evening. We want to have your questions, your reflections, your comments. So please don't be shy. Please share your thinking and anything you want to ask. Just to give you a little bit of housekeeping about how to do that. Please use the question and answer function that's in the Zoom webinar. That will either be at the bottom of your screen or at the top, depending on what device you're using. And all you need to do is type your question into that box. Don't worry if it looks like you're the only one putting a question into the mix. It is something that we're seeing behind the scenes as a feed of questions, and we'll be making sure that those are there for our panel to see and to respond to. So don't wait till the end. Put them in as you think of them. You can ask more than once. You don't feel you've got to store it up and ask your one quick question. You can ask things as they come to you, and those will be gathered up and brought into a mix, so we can ask as many as possible through as many interesting themes as we can. So I'm not going to be on screen very long. I just realised I didn't actually introduce myself. I just started talking at you, so I apologise for that. It's the heat and a very long day. So I'm Becky Gilmore. I'm from the Wellcome Genome Campus Public Engagement team, and I will be here just at the top and the tail of the events, but largely I'll be behind the scenes and picking up any questions that you might have. I will just want to explain how we'll work this evening. Beyond asking the questions, we'll hear briefly from our panel. I'll tell you a little about them in just a moment, but a large portion of the event is over to you. Coming in this evening, you can see so far we have around 120 people joining, and there's more I can see just coming in on the feed as I'm talking, so it's great to have so many of you in the room. In a moment, I'm going to bring up to our virtual stage our chair and our panel, but just to introduce them to you before they introduce themselves, our chair for this evening is Dr. Alex Lathbridge. Alex is a biochemist and a computational biologist, but you may have encountered him through his work as a science broadcaster, through his award-winning podcast, Why Aren't You a Doctor Yet? Very good question. But also on BBC Radio 4's Scientifically as well. Our panel this evening is a fantastic mix. I'm really excited to hear them talking together. Firstly, we have Professor Emma Thompson from the University of Glasgow. We have Professor Ravi Gupta from the University of Cambridge, and we have Dr. Chin Sen Wu from the Wellcome Sanger Institute. The Sanger Institute is based at the Wellcome Genome Campus, which is where we're coming to you from, at least virtually. So without further ado, I will ask our panel and our chair to take their place on the virtual stage, and I will encourage you to join me in virtually welcoming them to that. Thank you.
Wonderful. I mean, what actually works for clapping in this instance is pressing the raise hand and then lower hand icon again and again and again until you get carpal tunnel syndrome. Becky, thank you so much for that introduction. I'm your chair, Dr. Alex Athbridge. If you want to ask me questions, you can go with the he, him pronouns. I love them. And for anyone that's wondering, I'm wearing a sort of a nice blue shirt, and these headphones, they sort of make me feel like I'm in the 70s, which is a wonderful time to be because there was no coronavirus then. Much like Becky said, tonight we have an absolutely brilliant panel because, you know, it's impossible that for the last few months, if you haven't consumed any news at all, you might not know, but there have been a lot of new variants of COVID-19. And so the questions that is on the lips of like 99.999% of people is, what exactly is going on, right? What's changing? And then, you know, you have the follow-on question of, when do these changes actually become something to worry about? Now, of course, Becky stole my thunder because tonight we've got we've got a panel of brilliant people from up and down the country. That 0.1% of people that know the answer to the COVID questions. And they're going to tell us how genomics is helping us to keep one step ahead of the virus. One step ahead is all we have to be, with new tests and treatments. So here's how it works. These three wonderful panelists, they're going to discuss the science they do. And then after about 30 minutes of that, we're going to jump into general Q&A. So like Becky said, any questions that come to mind at any point in time, throw them in that Q&A box, and then we can jump straight into it. Now, without further ado, now that I've brought the energy up and I've hyped up these brilliant scientists, I'm going to start. I would like you to welcome to at least the virtual stage, Professor Emma Thompson.
Hi, well, thank you very much for the invitation to speak. My name is Emma Thompson, and I work at the MRC University of Glasgow Centre for Virus Research. I spent 20% of my time working as an NHS consultant in infectious diseases and looking after people with infectious diseases, including COVID-19, and 80% of my time researching various viruses. I used to work more in Uganda, but have been kind of recalled into working in the UK with COVID-19, and very interested, as are the other panelists, in viral genomics and using sequencing to understand a bit more about how the virus is evolving as it gets used to living in the human population. And so my research is very much focused on the sequencing. We also do work on looking at the likelihood of vaccine responsiveness by checking neutralization levels or antibody levels in people who've been vaccinated. And we work on, I run 4 vaccine trials in Glasgow. And if you happen to be in the Greater Glasgow and Clyde area, please sign up. And you're over 70 and you've had AstraZeneca, please think about signing up for the COV Boost study, which is one of our vaccine trials. Anyway, in terms of the genomics, I think you probably want me to go into a bit of detail about what we're doing. So we've used genomic sequencing to look at how the virus has evolved since it first emerged from Wuhan in China, and how it has spread across the world and eventually formed new variants. And so we're really interested in investigating these variants to estimate, for example, how many times the virus was introduced into the UK at the beginning of the outbreak, and we estimated that in a paper. And then we have been more recently much more focused on looking at mutations which might be, or changes in the virus which might be associated with compromising or escape from vaccines, so that we can try to keep up with making sure that our vaccines work for you. And yeah, I guess that's a fair summary. I can go into more detail if that's something that you would like me to do, but perhaps that's enough for now.
I mean, we always love detail. We love hearing about the ins and outs of your research because, you know, if you tell us a little bit more, a smidgen more, right, then the questions and the answers you give will be fantastic. And this panel will come together like a finely aged wine. I'm thinking about food a lot right here. So tell us a little bit more about what you do.
Yeah, so we, well, I can tell you about some of the research that we've done, and I can share a little bit of that with you. So firstly, we use genetic sequence data to estimate how many times the virus first emerged in Scotland. So I work in Scotland, and we've been doing a lot of the sequencing from people who've been infected across Scotland. And we've looked at how the genetic sequences look of the viruses as they entered Scotland. And we estimated, looking at the variation in the genome and then comparing that also with what we knew about travel history, for where people had traveled to when they became infected and so on. And we were able to estimate that the virus came in March 2020 at least 300 times into Scotland. And then there was a sort of brother paper, if you like, which was produced by other collaborators within the COG UK consortium, the COVID-19 UK consortium, which showed that the virus had been introduced over 1,000 times into the UK as a whole, which very much sort of tied in with what we found with the Scottish data set. And that I guess is important, because we may be at least partially responsible for informing government that introductions were very problematic and that they were occurring at high scale, and we were really being bombarded with the virus from multiple angles. And this was presented to SAGE, and I think that some of that has resulted in some of the travel restrictions that we're currently seeing, and certainly thinking about quarantine and so on is obviously clearly very important, since we're under such pressure from introductions of the virus from hotspots around the world. Of course, we export the virus ourselves as well, and the B117 variant originated in Kent in the southeast of England, and spread very rapidly through the UK during very high transmission, and was then exported around the world and became one of really the dominant variant. And so we've done quite a bit of work on that, and Ravi will probably talk about that because he's done work on B117. So the introductions is one of the sort of largest pieces of research that we've done. And then the next thing that we've been much more recently concentrating on is identifying ways in which the virus can change shape and avoid the immune response. And so we described, I think, probably one of the first what we would call escape mutations in the genome, which was N439K, which exists in the receptor binding motif, which is that bit of the virus that connects to the receptor, which is in our lungs, the ACE2 receptor. And we found that was able to change and avoid immune responses, and it kind of heralded larger changes that we saw happening in the viral genome in these new variants which emerged in the UK. And so those changes have now been kind of categorized as new variants, and there are 5 which are of concern in the UK, and many more of interest and under investigation. So we're currently looking in detail at the characteristics of those variants, and some of them are really don't look too worrying. Others look like they could eventually accumulate enough differences to mean that we might have to change our vaccine schedule. And so we're very involved with trying to sequence at high scale the changes that are occurring in the virus genome as it changes, and all viruses change within human populations over time. We're trying to figure out what those changes mean and whether or not we might need to change the vaccines. And I'm involved in working with the VUAG group in the UK, which is the vaccine update expert advisory group, which sits underneath the vaccine task force. And so we're very interested in that as well.
Wonderful. I mean, when people end their science with, is that enough? You know, there's really good things coming next. So, I mean, as you wonderfully said, halfway through there, next up, we've got someone who can shed a little bit more light into this, perhaps from a different angle, and bring in Professor Ravi Gupta. Now, Ravi's a bit on the spot because he joined the pre-Zoom call just a little bit late. So I'm bringing up his energy so he knows exactly where we're at with this discussion. Ravi. You are on mute.
Fatal error, and thank you. So thank you very much for the invitation to speak here, and it's a great honour to be on this platform with my colleagues. And so I'd like to just go back, I guess, to what I was doing before this all started, which was work on HIV, and in particular, we were interested in how we were going to get sustainable antiviral therapies to millions of people around the world. For the whole of their lives without experiencing drug resistance and drug failure. We know that HIV is one of the most mutagenic or mutation-prone viruses we know. And so that was sort of my training ground for what was to come here with COVID-19. So we spent over a decade working on drug-resistant HIV, how mutations arise, what order they arise in, what do they do to the virus, what do our new drugs do against those mutations, and how can we sort of limit progression of resistance at a global scale? So that's what we were working on. And when SARS, we thought this is interesting, but we'll carry on doing our own work. But eventually we realized we had to do something, and we got involved in diagnostic testing initially, actually, and that again was related to HIV because a colleague of mine had been working on testing for viral load to measure how much virus was in the blood of people with HIV rapidly, to sort of have a point of care test to do. From that, we then tried to go back to HIV, but then during my clinical period on the wards, we were faced with a sort of individual who had a chronic COVID-19 infection, and that he had been positive for this virus for something like 2 months. And he had a background of being immune suppressed. He was in his 70s. And I started thinking about the fact that this must be resulting in some evolution of the virus, because that's what we see in HIV. It's a chronic infection and the virus mutates. And I know that everyone had been saying that SARS-CoV-2 doesn't really very quickly, but I was still intrigued, and we started collecting samples over time from this individual, and then we tried a few different treatments like convalescent plasma and remdesivir, which didn't clear the virus, but when we looked at the genetic sequences, we found that when these treatments were given, and especially the plasma, that there was a great swing in the virus population dynamic, in that we saw lots of different mutations coming up across the virus, in particular in the spike protein. And then when the plasma sort of washed away from the blood system, because the individual didn't have any antibodies of his own, we found those mutations disappeared and different ones arose. And then when we gave more plasma again, some of those same mutations we saw previously came back, suggesting to us that the virus could bring or change itself to develop mutations or fix mutations that were going to help it escape from the and that led to really, I mean, this was basically the description of how variants arise, because of course, when we look at variants, they have multiple mutations, as we had seen in our patients, and some of them enable the virus to escape or to become less sensitive to antibodies that we make in response to a vaccine, for example. And we checked this, we tested this in the laboratory by making some of those mutations from the patient and then making them into an artificial virus system. And we found that the serum or the plasma that was used to treat him was really much less able to stop one of our lab viruses from causing an infection. So we managed to link the genetics from the sequencing that had been done on the individual, again, through the COG UK consortium. And we linked that to an actual phenotypic or a sort of character change of the virus in the test tube. And that was really powerful, because this was the first demonstration in the world, really, that the virus was capable of avoiding a block or a roadblock put in its way by mutating its spike protein. So that was really exciting. And that actually led us on to say to ourselves, well, let's look at some of these mutations that we found in the patient, because maybe they're in other viruses around the world. And you should recall that this is pre-variant. So, typically there were mutations coming and going in the world, and in the SARS-CoV-2 genomes. We were particularly interested in a deletion, a loss of 2 amino acids in the or 2 building blocks in the spike protein, that had been seen in our patient, and we found that that deletion caused the virus to replicate a bit faster or make itself more infectious. So we started looking at the database, of genomes that are compiled through something called GISAID. And that's kind of a resource that's been put together because of influenza originally, but allows researchers and scientists to share genetic sequences across the globe. And it's been instrumental in our understanding of this virus, and also the actions that we can take. So we looked for our key mutation, our deletion in GISAID. And this is sort of end of November last year, again, before B117 was reported. And we found a whole series of sequences, hundreds in fact, from the UK in GISAID that had our deletion in it, but had 7 other mutations in the spike protein. And this was shocking because, you know, you could see one mutation here and there, as I said, in the spike protein, but to find virus sequences with 8 different mutations in the spike protein, you know, we really sort of had to check that this was all correct. And, you know, it was a sort of jaw-dropping moment, but that was actually, that was us sort of, you know, identifying a cluster of infections that later was B117. So we went from one patient, exploring one patient in great depth, to obviously a new lineage that turned out to be a variant of concern because of all its properties. And that's a really important lesson. And both of those sort of discoveries or whatever findings were underpinned by the fact that COG UK had been set up by Sharon Peacock and various other people as an infrastructure. And we would have never got either of those insights without the genomic surveillance that had been set up. And so since then, we characterized B117 in terms of how sensitive it is to vaccine elicited antibodies. In other words, how well do the vaccines protect against B117? And more recently, we've been working with the Delta variant that originated in India, doing similar experiments. We're also very interested in how the virus behaves in terms of how infectious it is in cells, what are the mutations that do certain things to the virus, and of course, some of the biological characteristics of the virus and how it changes its protein structures and how it changes the way it decorates itself with the spike protein in order to achieve higher infectivity or transmissibility, or on the other hand, enables it to avoid the antibodies that we make to protect ourselves. So that's sort of the crux of what we do. And then we've done quite a bit of work in collaboration with them. For example, and we're finding that they're more proactive in vaccination and change our vaccination strategies longer term. Population. And I think I'll probably stop there. I think I've spoken enough.
Wonderful, because I was just about to jump in with, you're cutting out slightly, so that was a natural conclusion. Absolutely brilliant. We're getting so many wonderful questions in. So, I mean, if you do have questions that be answered, throw them into the Q&A box, not the chat box. The chat box is for us to talk about technical issues. So we come to the final panelist of the evening before, you know, the wonderful Q&A section. And so to round us off with this bit, I'd like to introduce you to Dr. Chen-Sin Wu.
Thanks, Alex, and thanks a lot for the invitation to speak here. So I'm Chen-Sin, and I work here in the Wellcome Sanger Institute. So in normal non-COVID days, actually, I'm a scientist working on developing technology in genome editing and single-cell sequencing field. So when COVID first started last year, around February, I remember we had actually a meeting at February 14th in Royal Society, and that's when all the scientists already start to talk about how to tackle this virus. So I remember there was 2 key things that was quite important. One is because the asymptomatic manner of the virus, how do we actually achieve population scale testing? And the second key point raised was even if with the vaccine, how do we actually keep monitoring the change and the mutation of the virus? So because with 2 points, at the point I was thinking maybe we should actually develop a test that allow point of care diagnosis, at the same time allowing a massive parallel sequencing to actually read out the virus sequence. So then I actually dropped out all of my original project and started this point of care COVID-19 diagnosis project at March last year. So together with a group of scientists here in Sanger, we developed a technology called Insight. So unlike most of the currently used tests, I think our tests have a few different features that's slightly different from the current one. So first of all, we want to use saliva instead of swab samples that give you a little bit of flexibility of how to collect the sample. And second of all, we don't want to use any kind of extraction method that have to manually done. We want to use a simple lysis buffer just to break out the virus. And then we also want to achieve isothermal, which is a constant temperature amplification of the virus for the diagnosis. And finally, like I mentioned, that we really want to have a test that have two stage, that the stage one is point of care that people can do at either workplace or home. And stage two is next gen sequencing, so that we kind of combine the advantage of both of these tests. Stage one will give you an instant readout, and stage two will actually help with the centralized data collection. And with a targeted manner, at least we can see some part of the virus genome by next-gen sequencing. So after a couple of months of work, we managed to actually put up our protocol and shared with the broad societies of scientists. And we also actually joined a global competition called X Prize. And with the given samples, from this competition, actually managed to get into the final as a finalist. And currently we continue to work on this project in testing the real patient samples, both from saliva and swab. And also at Weill Cornell University, we're collaborating with them and also test with more patient samples, hundreds of thousands of them, hopefully demonstrating the robustness of the assay. So that's basically what I have done in terms of contributing for the COVID.
Thank you so much. I did like you ending that with, that's what I've done. I mean, that's a lot. Now, I mean, not to put you on a pedestal above the other two wonderful panelists, but you've done a lot. So you can still now ask your questions, because we are moving into the general panel, the panel part of it. You've had a taste of what these people do, but now you get to interrogate them. You get to ask wonderful questions and have them answered. Now, just to soften all of you up from your ivory towers, my question is, we've had so many different names thrown at us. Like I've got a few here. Your B1.17, the Kent variant is now Alpha. Your B1351 South Africa is now Beta. The P1 from Brazil is Gamma. The B1617.2, the India is now Delta. So with all these different variants, I think the one thing that I need at least one of you to answer in a very simplistic way is, what actually is a variant? If one of you doesn't jump in, I'm going to pick you at random, like we're back at school.
I think it's that, or do you want to do it, Ravi?
No, you got the first.
I guess the simplest way of explaining this is that so a couple of key terms. So mutation usually means just one change in the genome, which is important because it might change the shape of the virus slightly in one place. But the thing about the new variants is that they contain many mutations, and we think about these as like constellations in the stars, that they come with many changes. And we noticed this happening at the beginning of last year, that there were suddenly popping up variations of the virus which contained, or variants of the virus which contain multiple mutations. And so a variant contains several key mutations, and these are laid out, and we've made laws for them in terms of which changes are characteristic of each variant, whereas a mutation just refers to one change. Does that make sense?
That makes a wonderful amount of sense. So, I mean, this is open to all the panelists here, but in the last, we've been at this for, a year, if not longer. I I mean, for each of you, what has been the biggest moment so far in our research with COVID-19? I'm going to start with Chensin.
So I guess the biggest moment for me per se is when I saw my technology work, and it's very comparable in terms of limit of detection compared to the public health implant method. So that for me is a kind of a big moment for me per se. But personally, I think it's the moment when I see that the senior people that was queuing up for the vaccine because of the sequencing or all of the scientists working towards this goal, for me, it's actually quite a touching moment to see all of them actually getting it.
Ravi, what about you?
In terms of the biggest moment sort of for the field, I mean, I guess a couple of things. I mean, one was the demonstration that vaccines have, are highly effective. Some of the trial data that we were all waiting for. And then I guess the data from steroids, dexamethasone, for example, in the treatment of COVID-19 disease. Those are the big moments, I think. For me personally, in terms of our research, as I mentioned, one of them was kind of stumbling upon B117, as a total, accident. At the same time, of course, PHE were investigating this big outbreak of infections that were defying logic. And they had, identified along with COG UK that this was a new variant. I mean, that was just amazing because we just didn't know that this could happen, and to have come from a patient where we could see it happening to it playing out in real life within weeks. I mean, this is the irony that this was just a coincidence, this one person and suddenly seeing the whole explosion of a new variant. It was really sort of strange, that it coincided so closely. So yeah.
Wow. And I mean, so you, Emma, what would you say to round it off?
I think scientifically, the biggest moment for me has been the emergence of vaccine technologies, and specifically the RNA technology, because you can literally look at the genetic sequence of what we do, all three of us, and then you can program that into a vaccine now. And it's a huge revolution in vaccine technology. So instead of having to spend a very long time producing protein, that you do with more traditional vaccines, you actually inject a recipe for that protein into someone's arm, and then the protein is manufactured, kind of using their arm as a factory, if you like. And that means that the whole process can be so much faster. And I think it's extraordinary, and I think it's going to revolutionize vaccines for other diseases as well. So for me, that's the A1 scientific discovery. And I guess personally speaking, the kind of highlighted moments have been when I was called by Sharon to join the first meeting where we decided to set up COG UK. And there were a number of us who do sequencing from around the UK, and I just remember the feeling in the room when we realized that this was going to be something quite serious, and it was going to wash over us and change our lives. And I think it put a shiver around down my spine actually at that meeting. And that was it was really the foresight of Sharon, who's been a great leader of this consortium, and also the funders who were there, so the funding agencies were there as well, and invested very quickly so that we would be able to track the virus. And I think that was a really important moment.
Okay, wow. And so, I mean, just jumping into the questions that are in the Q&A box. I mean, starting off, one really interesting question, shout out to Brian and Gwen. Is there a danger of ever accidentally creating a variant when doing research to combat the virus? Now, it's a big question. Please, someone feel free to jump in, preferably one of our panelists.
Yes, I mean, that's something we've been talking about very recently on some of our calls. I mean, there are, with variants, we are infecting cells, infecting animals, in fact, with variants. And And the question has come up as to, oh, could one be creating conditions for something to change, within the lab? And I think that everyone has taken it very seriously in terms from the beginning that we're going to, be very cautious and about what's being done. And that's an ongoing discussion. But I think the key thing is, I think the scientists involved are highly able and discuss their work with groups of scientists in order to arrive at a consensus of what we should and shouldn't be doing. So I think it's an important question, but I think the risk of creating something in the lab that's a sort of, a VOC that's derived from a lab, I think is minimal, because of the checks and balances that we have.
I think we'd have to make an effort, wouldn't we, to make something really nasty in the lab. There are quite stringent health and safety regulations that we have to follow, particularly if we're thinking about modifying genetically viruses. We have to submit that to a national agency to be approved. And then we also have to have very stringent measures in our laboratories. If we are growing live virus, for example, we have to have very high level biocontainment. But it would be possible. I mean, of course, and we have to be aware of that, and we have to take great care, and we do.
Okay, Chenson, anything to add on that?
So normally I engineer the human genome, not the virus. I'm really not an expert on this particular point. But but I guess by the end of the day, it's the evidence from the sequencing data and all of the evolution, including other type of virus comparison. I think we can reach a final conclusion, but I think the science will speak itself.
Okay, I mean, now, talking about variants, being accidentally created in the lab. Now, one of the big questions we've had in the Q&A box, this one I'm looking at from Neil, was the virus created in a lab? And if not, what animal did it come from? Because, these are the questions that people have been asking recently. It's been up and down, depending on where you get your media from. So it's something that, to you experts, I'd like to ask, was the virus created in a lab? And if not, where did it come from?
I think the word, just to start that, I mean, the word created is I think itself not the right term. I think that the way to say this is, did the lab come from a natural origin in the wild, or the interaction with humans, or did it emerge from a scientific facility or a man-made facility? So I think that's the way to phrase it better, so that you take out the whole manufactured, or I don't think anyone has had the goal to make something intentionally that would cause harm. Of course, it's been done in the past with bioterrorism and and various other sort of branches, but certainly with viruses, they're so unpredictable that I think it's not the first thing you'd go to if you wanted to do damage. So whether this was a lab leak was initially discredited partly because I think President Trump had aligned himself with a lab leak conspiracy theory. And so I think a lot of scientists, including myself, were not able to discuss the fact that we weren't sure. And there were two possibilities, and we should treat them equally and disprove them. And that's what we do as scientists. We disprove hypotheses. We say, is it this or is it that? And we say, well, it's not this and it's not that. But no, and so that's what we wanted. The WHO mission didn't necessarily achieve that because it wasn't allowed to treat both possibilities equally, let's say. And so I guess that's where some of the controversies come since then. And I think that, for history to say we're not sure is okay. But I think a lot of people don't think it's okay to say in the history books that it was definitely an animal transmission, because we don't know that yet. I think that's, and Emma's probably have some views on it. Yeah,
I do. I actually agree with you, Ravi. I think I wouldn't like to overstate what we can tell through genomics. So I think that in terms of proving or disproving whether or not the virus came from a laboratory, we can kind of contribute. So we can tell you that it doesn't look like an engineered virus. So if we were going to, Ravi or all three of us were going to get together and try and manufacture a nasty virus, doesn't look like that. It doesn't look particularly like viruses that have been sequenced before. So the sign, if it had come from a laboratory, the sequences would have had to be kept secret, and any kind of changes that might be associated with trying to make a virus sort of transmit in the human population, they would have had to be completely unpublished. So I think we can say it doesn't look like an engineered vaccine with kind of known bits of viruses that have previously been described. I think the thing personally that's difficult to really for us to be able to answer is whether or not the genetics can tell us anything about whether or not someone might have been using bat tissue or something like that in the lab, and then didn't sequence it and did it escape from there. That's possible, although probably quite unlikely compared with the likelihood of a transfer from as a zoonosis or a jump from an animal into a human. For example, in a marketplace where there are many species in very close proximity to humans. So in terms of the likelihood, it strikes me that the likelihood is much higher that it's an animal jump. But I don't think that we can completely disprove the laboratory theory through genomics. We can say that we can't see evidence of it, certainly.
So I think I just want to highlight it in a sense that for scientists, it's also hard to judge whether it's come up from the lab or not. I mean, even if we have genomics as a powerful tool, but if you imagine how would scientists actually figure out the origin, still they look at the genomic sequence of a virus, which is a stretch of A, T, C, and G. They look the similarity between what we sequence now and what actually sequenced before in different type of animal. They use different algorithms to compare the similarity between them and try to generate hypothesis or conclusion of which one is more similar to another. And from there, they try to draw a conclusion. But even with all of this, so you could have the risk of not to sample the particular species that they might originate from. In fact, if you think about the SARS, original SARS that happened decades ago, actually, scientists were very much wrong at the beginning in terms of the origin of the species. And after many years, they figured out actually it's not originated from the cives, but rather from bats. So I think it's a very difficult question, and it's also difficult for scientists. Like it's hard to draw an absolute conclusion, even with genomics.
Okay,
it's a bit like the kind of pink teapot circling the Earth, the Russell kind of comparison, isn't it? If you don't have a telescope powerful enough to see the pink teapot circling the Earth, can you disprove it? And I think that's really the difficulty that we have, but we certainly haven't seen a pink teapot circling the Earth, if that makes any sense.
Well, jumping off of your willingness to talk about pink teapots and whatnot, we do have a question aimed directly at you, Emma. Now, someone wants to know that if you could explain in a bit more detail in what ways would a vaccine, in what ways would a vaccine need to be changed in order to overcome different virus mutations or variants?
Yeah. So, well, the virus has accumulated many thousands of mutations already, and that happens with all viruses. So, influenza or just common cold viruses and so on will accumulate changes. And most of those are actually not very helpful for the virus and often detrimental. But now and again, one mutation or two or three or four start to pop up where they give the virus an advantage. And I noticed in the chat there was a question about why that happens, and the reason why that happens is because the machinery that the virus has for replicating itself doesn't work very well, and so every time the virus divides, a mistake is introduced, and so usually those mistakes are not good for the virus, but now and again something happens that, for example, that would cause the shape of the spike protein, which is that projection out of the virus particle that you see sort of spinning round, and it's really important because that's what our antibodies recognize in order to kill the virus. And so if those change shape, then you can imagine that that you don't get the interaction any longer between the antibody and the virus. And so we can see that from the genetic sequencing. And I think this is probably where the great power of genetic sequencing lies, that we can actually see when there are changes arising that we might predict might affect a vaccine. And then that needs to be backed up by experimentation. So we need to be able to show that in the laboratory by doing what we call neutralization assays, which is to test virus particles with people's antibodies who've been vaccinated and see if the virus is prevented from getting inside cells. And so that's what happens. So gradually the virus accumulates changes, and eventually that results in the vaccines not working quite as well. And it's probably a fairly gradual process, so it's quite likely that the virus needs to acquire several changes to start to escape from antibodies. And so we can track that with the genomics with our sort of early warning system that we have through COG UK. The experiments can take a bit longer, and then of course the clinical trials take even longer than that, but if that's the #1 data. So if you start seeing that people who've had vaccines are getting infected, that's a real problem. And then you can tie in, you can figure out why that's happening by looking at the changes in that, and usually in the spike protein part of the virus, to see if it's, you can nail it down right down to the sort of building block of a protein, the amino acid level, to figure out which changes are causing a problem. And then once you have that information, you can program that into a vaccine. So like with that mRNA technology that I was discussing before, you can actually pretty much print the what's needed in terms of the genetic sequence of the virus, literally, and put it into a vial and purify it and so on. But that then becomes the next vaccine.
Okay, and just so you know, I understand it, because all three of you are talking at very high level here. And as someone who doesn't work on this day in and day out, sometimes I can struggle to understand. And so, you know, myself, much like Lucy in the questions box, you know, wants to know, can you explain really how this mRNA technology actually makes a difference in this process from, say, you know, what we consider like regular vaccines? I mean, how does this mRNA, RNA technology make things better? I hope better. How does that work? I mean, Emma, Ravi, Jensen.
I can answer briefly, and then maybe they want others, or do you want to go ahead?
No,
it's
please.
To do with time. I mean, so to grow protein, it just takes a long time if you do it in a laboratory, and it has to, also is a very long sort of process. So if you have a big factory for making protein vaccines, which would be the much more standard. So this is, you have to remember that there's RNA or RNA which gets it's a sort of code, it's a recipe, if you like, for making the building blocks of the bit of protein or the bit of the virus that you're going to expose people to in your vaccine, so that you trick their immune system into thinking that they've been exposed to the actual virus. And making that protein takes a lot longer than making the mRNA. There are other reasons why vaccines are becoming faster as well, but that's one of them.
So just to give you like a very, very brief concept of what this is mRNA protein. So if you think about the virus itself is RNA, and RNA is basically also 4 letters. So after we sequence, we know those letters. You almost use with some kind of modification, but almost exactly same letters in this RNA vaccine, which is part of the virus. It's only on the spike region. So the reason why this is so simple, adaptive, and quick was because this whole mRNA production process relies on a single reaction called in vitro transcription. So basically, we assemble like a group of machinery that normally happens in our cell or in virus, quickly actually transcribe the DNA to RNA in an enzymatic manner. So this process is very fast and very abundant. So because of this, also because of the great technology development in the nano particle field, so after you generate RNA, you simply just wrap them with these lipid nanoparticles. So this will be enough to actually get the mRNA delivered into your cell. So because of all of this, it's very easy to change. If you imagine if the virus become mutated, you can just change the letter. It's as simple as that. So it becomes a very kind of attractive technology in terms of vaccine.
What's great there is Ravi's unmuted himself.
Yes, you want to? I mean, in terms of what to add, I mean, Emma's discussed the fact that protein-based vaccines, obviously, you know, they're harder to make and purify, and it's easier just to send the message for the vaccine in rather than the actual protein. There are other vaccines that do something not so dissimilar, like the AstraZeneca vaccine, for example, also sends a message into a cell, but it's a different type of message. It's a DNA, and the DNA is in the form of an altered virus. So it's doing something similar. So we're still not really sure as to why the mRNA vaccines, you know, they may generate slightly higher responses, but I think the data is still emerging as to what the differences are between them. But what's fantastic, I think, is that we have a number of different ways of doing it, and RNA vaccines may not turn out to be, you know, the best in the longer term. We just don't know yet which one will be the best for us, and therefore having a range of different approaches is really important.
Okay, wonderful. I mean, now away from vaccines and back to variants. Now this question's really good, so good that I've gone over it twice, missed asking it to you, and then come back to it. It's like Christmas morning when you think you've opened the present, but then you haven't, and then you get it there. But without further ado, Denise has this wonderful question. It is, you know, we've mentioned all these variants, you know, your alpha, your beta, your gamma, your delta. Now, is there data worldwide where it lists the most dominant variants in each country, you know, from the UK to the US, Brazil, China? You know, is there a place where scientists, researchers like you can go and you can see, you know, all the variants that are present in this place and which is the most dominant?
Well, yes, to some degree, yes, because I was waiting for Emma to answer there, but you can to some extent, but of course it depends on how no, I wouldn't say how good the sequencing is, but how, you know, how much investment there's been in different countries to do genomic sequencing of positive cases, and you know, the sequences go into GISAID, and so you can use GISAID, for example, this database I talked about, to see what the dominant variants are in different countries. But the caveat is that, of course, a country that doesn't do any sequencing has no variants at all. So the more information you get, the more understanding you have of what's actually circulating there. That makes sense. But yes, many countries have dominant strains. There's no doubt that certain strains do dominate different countries.
I put the link for Denise in the chat. If you want to take a look, that's the website that we look at.
Wonderful. Okay, I mean, just to jump onto the following question here, because, you know, if you go on GISAID, you can see, I keep having to say that very, very, very firmly, GISAID, you can see all this information. And I mean, for people that have looked at this information, who understand this, I mean, are the variants that we see, are they occurring at a constant rate, or are there clusters around the world where there are higher rates of variation? And if there are, why is that happening? Is there a pressure there?
Mm-hmm. So I'm going to start with that. I could take that one. The virus was mutating relatively slowly, as far as we could see, until the end of last year, and then suddenly we saw quite a bit of a step change in variations, which had become fixed in the population, with multiple changes, multiple mutations forming these new variants. And almost certainly they don't occur just everywhere. So they tend to occur in places which have had very high rates of transmission. So those areas have been places like the UK, India recently, Brazil, for example, and South Africa. And when there's very intense transmission, the virus induces immune responses in a significant proportion of the population, and that may drive change through kind of similar responses, or what we would call public antibody responses in the population, which might drive the virus in the same sort of direction. And so I think there was another question in the questions about whether whether vaccines might drive change, and they will do. And so but also exposure in the population would drive change as well. And then Ravi has a key paper where which he described earlier, which describes change in someone who had chronic infection. And we've seen several cases of people who are chronically infected. It's a really uncommon clinical sort of manifestation of infection, but in some people who have very weak B cell responses predominantly, so antibody responses, they can be infected for many weeks, and that may cause changes to arise in the virus in individual people, which then might transmit differently.
Okay, wonderful. Thank you for that. I mean, this is me sort of now combining a couple of questions that we have. Now, this might be good for Chen-Hsin or Ravi here, but when we're talking about all these variations that are happening, do we really have an understanding of the factors that drive the success or the failure of the evolution of variant? Is it transmissibility, or is it lethality, or it being able to compromise the immune system, or is it like a random process? I mean, what drives a variant becoming a variant, and a variant becoming a successful variant?
Okay. Well, it's natural selection at the end of the day. That's the way that these things work, and we see evidence of it all around us. And we're the evidence. We're the result of natural selection. Organic entities changing, making mistakes when they copy themselves and have children or daughter cells. There are mistakes made all the time, and some of those mistakes give an advantage to that organism, and some of them are bad for the organism. So the ones that have good changes survive, and the ones that have bad changes die off. So that's how the variants emerge. So the SARS-CoV-2 is making mistakes all the time, and it's making huge numbers of particles. I think this is the other thing that we haven't really raised. I think the actual turnover of this virus is so immense. It infects and produces baby viruses very, very quickly. Within 6 hours, let's say 6 to 12 hours, it's already made copies of itself after entering a cell. So it's doing that at huge pace, and therefore the number of mistakes that are being made is quite large. And so if you can imagine this going on in one person over days, weeks, as we observed, what's happening is that the virus is making lots of copies of itself. It's got an immune system that is kind of trying to destroy it. It's trying to get around that immune system. And by making lots of different varieties of itself, some of which have an advantage, those advantageous ones will survive as the antibodies and all the immune defenses come in. And those guys will then kind of take over the population in the patient. And that virus is adapted. It's learned to deal with the immune system. And as Emma said, a lot of us make very similar responses to each other. So when that virus has done its training in patient X or me, it jumps to you, and it's done its training, and it's now better at avoiding your defenses because they're similar to mine. And that's why it becomes a variant, because it's got new mutations. It spreads better to you. It makes you sicker maybe, or it makes a higher number of virus particles every day. It then transmits from you better to someone else, whilst avoiding antibodies and vaccines. If that person's vaccinated, it might even infect them despite the vaccine, because it's got enough mutations to get around. Luckily, at the moment, we're in the situation where variants don't cause death when they escape from our vaccines, but it may not last forever. So we do need to be vigilant. So that's, I think, how the process is working. And then there's this thing called recombination, which we're not really entirely sure of right yet. There's some work going on where two viruses infect a cell, two different variants can come into a cell, two different varieties of a virus. they can swap genetic information with each other, and then you get one that comes out with a sort of a mosaic or a mixture. And we saw that in HIV all the time. It's an incredible sort of way of a virus changing itself dramatically through a very simple mechanism in a way, just by swapping between different viruses. And that's probably another way that the virus becomes more diverse and enables itself to get to be better.
Chin-Sin, anything to add to that? What? You said my name.
Not really. I think both Emma and Ravi give a lot of insights on this. But from a relatively outsider point of view, it's just I was also trying to see like exactly how do people actually define what are the variants that actually causing concern, or what are the variants are perhaps just genetic drift that they will float away with evolution pass by. So I think generally, although you heard about all these different variants, in fact, there are 4 of them that I think now the world has been classified of variant of concern, which possibly could influence, such as the vaccine neutralization capability, etc. And there are 5 of them are variants of interest that have less of an impact. But I think, but in reality, actually, there are more variants that's going on, but it just probably not being naturally selected, therefore it didn't cause any impact in our scientific research, etc. But yeah, but variance is something normally happens, but it's just not every single one will be very important.
Okay. I think that about covers it, at least here for that question. Now, just to let everyone everyone in the audience know, there are a few questions that are coming in on the topic of symptoms and susceptibility. Now, these aren't being ignored. It's that next month's event is entirely on that. So if you come back there and ask these questions, you'll get a far more fantastic answer. I mean, all the panelists here could obviously give very good answers, but next month, amazing answers. Now, I mean, one question here from Nick. Now, we have moved further and further into our journey with COVID-19, the SARS-CoV-2 virus. Now, there have been reports that a high percentage of the UK population have COVID antibodies. Now, if this is true and the whole world can reach this level, assuming that we get some level of healthcare equality around the world, could it be possible that the coronavirus could be eradicated?
That's a difficult question.
That's why we put them to you. Thank you, Nick.
Right. My answer is going to be no, because of the fact that we have the common cold viruses as an example. Of course, we don't have vaccines to those, but despite the fact that we become partially immune to those viruses, they still kind of hang around and circulate and reinfect us at different seasons. And SARS-CoV-2 will probably do the same thing. Of course, we have vaccines, but we can already see that the virus is adapting to vaccines. And if we could vaccinate everybody straight away, we could probably make it extinct, potentially. But because we can't give everyone a highly effective vaccine and monitor it and do it all in one go, we end up with a patchwork of vaccination around the world, and you have pockets of people getting infected and transmitting, getting these long-term infections that we discussed. And I think that's the key. The key barrier to this virus being uneradicable, if that's a word, is similar to HIV in a way, in that it causes a chronic infection. It becomes very diverse. It can then infect lots of people. And that's what enables it to persist in the human population and is very difficult to extinguish, because we also have the problem of what we call asymptomatic infection, where you can be infected and not even know. And so with all those factors combined, this is not a disease that we're going to eradicate, especially with the inequality that's going on about vaccines, problems with production, the fact that vaccine-induced immunity is probably not going to just last forever. We'll probably get people with a peak and then it will come down over months. There are obviously the elderly, the people who don't respond to vaccines as well. They're more vulnerable, and so they may be sort of reservoirs of infection. So I think, unfortunately, it's a bit of a no, but it doesn't mean we can't control it and live normal lives.
I could add to that maybe, but I agree with everything Ravi's just said. The other thing is, of course, that the virus still exists in the probably in an animal reservoir, and so do many other similar viruses. And I kind of caught, and there's so many questions, but I did catch in there that, you know, what about SARS-3? And unfortunately, we're pretty confident that there are SARS viruses existing in bats, which may have the potential to jump into humans, and so there's the possibility, there is the possibility of more viruses which are related to this one jumping in as well. And so we need to think about the way that we live our lives and why these things are happening. We've slightly lost the kind of balance perhaps between living in the planet that we occupy. We're intruding into parts of the planet which previously have been more kept for other animals, and I think that this means that we'll see more jumps of viruses into the human population. And the other thing that we're doing, of course, is that we all go flying all over the place all the time, and scientists are definitely no better and probably much worse than the average anyway, you know, someone doing another job. We do a lot of traveling. We could probably be doing Zoom a lot more, and it would be better for our planet, and we're less likely to spread dangerous viruses amongst the whole global population.
So I think I agree with Ravi and Emma have said. I was thinking actually before, like in terms of the virus that completely eradicated from this world, smallpox is a good example. By the end of the day, it doesn't exist anymore. But then compare the smallpox to the current SARS-CoV, I think there are a couple of differences make it probably hard to eradicate. One, the smallpox is double-strand DNA virus, so the mutation rate is relatively lower, and the host of the smallpox is human, mainly human, unlike the SARS-CoV, which they can transmit between animals. So if you imagine, even if we eradicate between human, what about the animals? Can they actually get back to us when they evolve to a certain extent? So I think by the end, to completely eradicate is probably hard, but at the same time, if they just become like a big flu pandemic, such as the current flu, perhaps it's just not as severe as what is happening now. So that's my hope that what will going to happen.
Okay, so these are really interesting answers. And I guess there's another question that sort of piggybacks off of that. And I'm not answering this just because the person who asked this just followed me on Twitter. But like, what is the likelihood that we could get a variant that emerges that goes around our current tests, or could vary so much that it's considered like a separate coronavirus? Could we ever have that sort of two coronaviruses going out there in the public, if that makes sense?
Wait, do you want me to take that one? I could start on that. I mean, I think, yes, it's absolutely possible. And it might happen as a result of a jump from an animal host, so another outbreak, or it could happen as a result of changes in the virus that we already have, changing in different directions in different parts of the globe, for example, under different selection pressures. And as Ravi said, it's a simple sort of evolution, which is the fundamental basis of biology, I guess. So if the virus has an advantage to transmit, it will take it, and those advantages might be different in different parts of the world. But so yes, definitely possible. It's a very semantic difference. So the difference between a variant and our mutation is one change. A variant might be 17 changes. And then a change from one SARS virus to another would be a whole lot more. So perhaps another SARS virus might be only about 1/3 the same. So these are sort of, it's all the same process. It's just a matter of how far it goes.
I assume Ravi's about to bring up the rear with that.
Well, I know, as Emma sort of put it very nicely, that that anything's possible. In other viruses that are changing this quickly often get subdivided. You know, for example, the HIV, you have different subtypes. Same with hepatitis C, we've divided them into different lineages, and that's what we're doing with SARS-CoV-2, but it's happening quite fast with SARS-CoV-2. So things are going to get complicated quite quickly, I think. Or they already have. So, you know, and that's why we're having to come up with all these new naming systems. So I think it's going to be very exciting, but very confusing. And I don't think we've ever dealt with anything like this before. And I think it's hopefully arming us with the tools to deal with whatever comes next, because there will be something next unless we either put some mechanisms in place or whatever. But I think that a lot of important learning has come from this.
Okay, and I'd like to remind all panelists to switch off their phones or any WhatsApp that they might have open. This is the most important thing right now, the questions that are being asked here, and you're here to answer them, all right? So one question that is here is, you know, once we actually have the virus, let's say the virus gets into the body and it infects a human cell, the virus starts to replicate, right? How many actual virus particles get released out into the surrounding cells in the body? Oh, it's a specific one.
That's a hard one, because I mean, I don't know what Emma's group is also doing live virus experiments, so are we, but we don't really quantify the number of viruses coming out, and it's really hard to do it. I mean, you could potentially calculate it, but we do a lot of our work in cell lines that are kind of, they're cells, but they're human cells, but they're kind of modified. They're cancer cells. They're not really the sort of cells in your body. It is very hard to come at that number, but it would be good to do that. I think that as we go forward, I would like to see more endeavor in understanding the turnover rate of the viruses, answering the question you just did. Some of these fundamental things that really we should know by now, but I think it's a big challenge to do it.
I'm trying to do a fast calculation in my head of how many viruses one person might have. It is a lot. Like
I quite like that. I quite like that Ravi gave us a minute and a half, like, nice way of saying no. And you're there in the background with, actually, Ravi, you're wrong. I did it on a piece of paper, and it's actually 20. But I mean, how
it's
many?
millions. It's a lot. It's, when you look at if you quantify the, you'd have to, it might take us a little bit of time to get the calculator out and calculate it. But like, some people have got, viral loads that are really, really high, and then it varies actually from person to person. So you get these different sort of viral loads, but you get into the sort of spectrum of like 10 to the 6, right, Ravi? 10 to the 6, like in a very small volume of spit. And so then if you think about how much spit you have, I'm not sure how much, but you would multiply that. And it's a lot, millions of virus particles.
I didn't. Now you've got me quantifying the amount of liquid that I hold in my mouth, which is very strange. It's like when you tell someone you are now breathing manually, which is what 115 participants have just heard. Now, I mean, we've done a lot of research into this virus, starting last year, and now we're at the point where we have vaccines. I mean, going forward, are vaccines likely to hinder mutations, or are the vaccines that we currently have more likely to encourage further mutations and sort of variants forming? Now, maybe Chen-Sin, you might be good at this one, because I'm thinking about sequences, I'm thinking about sequencing.
Yeah, so I think vaccine is to a certain extent putting a pressure on the virus evolution. So it will automatically kind of pressure it to actually further mutate, especially at the place where the vaccine actually targeting. That's why all the scientists are talking about the spike region. And I think scientifically, you can also see the spike region is mutating relatively faster, like compared to other regions. So yeah, so I don't think it's facilitating, make them change faster, but rather putting a pressure to actually select those ones that actually can survive with the vaccine.
I mean, Emma, I see you off mute, so I'm assuming you're going to answer the question. I'll put you on the spot now.
I actually slightly didn't catch the total question. Can you can you tell me again?
That's okay. So basically, are the vaccines that we currently have, are they more likely to hinder the formation of variants, mutations, variants, or
I think
encourage
so. Yeah, no, I think the balance is in against the virus with vaccines for sure. So I think they will hinder the formation of new variants, and that's because new variants are arising in areas of very high transmission, and we're starting to see clearly that not only are vaccines hindering transmission, but the important thing that they're also doing is breaking the link between infection and very severe illness and admission to hospital. But they are reducing transmission, and so that probably reduces the chance of getting many more new variants. Having said that, though, there is a pressure, there is a selection pressure, which the virus has to try to get around. And if it can't, it will die out if we vaccinated everyone, for example, as someone has had earlier suggested. So I think it's all, I think the vaccines are definitely going to help, but they may not be just quite good enough to eradicate the disease.
Yes, completely agree with that. So yes, the balance is certainly in favor of vaccination, because the big driver of the diversity or the mutations we've seen has been chronic infection, and chronic infection is very rare, but the more infections you have, the more people you have with these long-term infections. So the way to deal with that is through vaccination.
Okay. I mean, we've got time for just maybe a couple more questions. So I'm just pulling one out again. So, I mean, a question that someone has. So around the time, a question from my brother. Now, at the time, I had my first AstraZeneca dose and some side effects after a few hours receiving the jab. Now, how could we explain that the vaccine can elicit a response in the form of side effects within a few hours, whereas with the real thing, the virus, when you're infected by it, the symptoms don't develop before, day 2 of the infection up to, much later, if any at all, talking about the asymptomatic cases. So why the difference there between side effects and the effects you actually get from having the virus? Gonna pick on one of you.
Okay, I was going to volunteer Emma, but now I'm feeling guilty. So, well, the main thing is that, of course, a reaction to a vaccine is potentially just because you've had, something injected into you. And in the case of an mRNA, you said AZ just now. So there are things in that vaccine that might trigger a reaction. So, and it's not the virus. The virus hasn't been made yet. So there's the code for the virus in there, but nothing's been made yet. So when you get a reaction within hours, it's more likely to be just the fluid that's in there, the stabilizers, maybe even the DNA that's in there, causing a reaction. And of course, that's why the infection itself obviously takes longer to have any effect, because you've got to get the virus infecting cells to cause a problem. I think that's the short answer, isn't it? Probably has something.
I could say something more. I suppose. I mean, the side effects of the vaccine generally are not the same exactly as the virus, and maybe when you do have replicating virus at kind of low level, you do get minor symptoms, but you kind of dismiss them until the viral load is at such a high level, and you've got such a lot of inflammation in your lungs that you notice it, and you don't need all your lung function, but just like you don't need all of your liver or both your kidneys, and so the virus can probably reach a certain level before you really start to feel very unwell, and that's quite different from how you feel with a vaccine. I think, you know, as Ravi says, there are components of the vaccine that you might have a slight reaction to, but also the side effects you get with the vaccine are not really on the scale that people who are symptomatic with COVID-19 get. Some people are asymptomatic, to be fair, and that is also quite an interesting question. Why are some people asymptomatic with quite a lot of virus around? And I guess it's because they moderate inflammation in their body in a different way for some reason. It tends to be younger people and children are, you know, the people that have high levels of virus without many symptoms. It's probably not very fully explained, actually.
Exactly. All three of you are fonts of knowledge, and I imagine we could go on and on and on until we could be here until like 11:30, and people be like, "Oh my God, did you go to that Sanger event?" Yeah, it just kept going on and on. It was so fascinating. I couldn't look away. We do have to bring the event now, at least the panel discussion, to a close now. I'd like to thank all three of you, Ravi, Emma, Jensen. This has been fantastic. So I'm now going to pass it back to Becky Gilmore. So if we could all shut off our videos.
Thank you, Alex, and thank you to our fantastic panel. There was a huge amount to take in there. I know I've left even more questions than I had before I came in, but I hope you all enjoyed this evening. There's still so many questions that we could have got through in the conversation box there, and so my apologies if we didn't get to yours. We tried to capture as many around different themes as possible. As Alex mentioned, we have another of these events happening next month, Wednesday, the 14th of July, same time, 6 p.m. Here on our Zoom channel. That will be looking at the concept of artificial experiences of symptoms. You know, why is it that someone could be completely asymptomatic, as the panel were talking about just then, through to obviously the very sad loss of life that we've experienced here in the UK and indeed around the world. Our panel will be exploring that in much more detail, and our chair for that event is Dr. Angela Saini. So I do hope we'll see you back here again for that. I will draw us to a close in a short moment, but just to flag that at the end of the session here, you'll see a link pop up, which is to a very short survey on our SurveyMonkey account. It would be fantastic to have your input of what you think of the event. It really helps us shape what we're doing, making sure our programme is meeting your needs, your hopes of what you want to get out of these things. So if you have a few minutes to spare, then please share your thoughts there. If you don't have a chance, you'll receive a link through your booking email as well, so you can always answer it at a later date. In the chat box within the current webinar, you'll see some links to some of the things that have been mentioned this evening, and also to some of the sites for to get more information about the COG consortium and the work that's happening around the UK at the moment, and also a link to find out more about our events, not just the COVID connections, but other ways that you can get involved with the things that we're doing here at Wellcome Genome Campus. So I would just like to end and say once again a huge thank you to our panel and to our chair, Alex, and I hope we'll see you all again very soon. So thank you all for being here and have a lovely rest of this evening. Thank you very much.